So what we're going to do today is I will spend most of our time doing the hands-on, and what I expect you to do is not just be able to follow along, but just you don't have to wait for me. Proceed. The notebook is available on this Quarto on this Google Drive. It's a Quarto notebook, and by now I guess you all know how to run Quarto notebooks. I'll just give you a very brief gist, which I had started last time, about what is the problem about. And the problem is that somebody looked into mice and the Alzheimer's version of the mice. The question that they were interested in is what was known previously is microglia are a very special kind of are one of the cell types that exist in the brain, and Alzheimer's disease is known to be associated with microglia. What is also known that if people have variations in their TREM2 gene, TREM2, then it can prevent development of that disease, or it can actually ameliorate overall what happens. So if you look into this data, what they try to do is they took a knockout. So they knocked out the TREM2 gene and then try to mechanistically understand that why does it happen, like what processes is TREM2, because if you have variations in TREM2, then it can actually help you prevent the onset of the disease. And basically what is happening is it's preventing the accumulation of the tau proteins, the alpha, the amyloids and the tau proteins overall. The question then to understand is, if I knock this gene out, what changes happen in my system? Because if I understand that, I will be able to mechanistically understand what is the function of that gene. Right? So microglia are like the immune cells of the brain. They try to do like they consist of multiple different cell types, but one of the primary goals is to remove things which should not be there, and that's kind of the function overall that are they carrying out in this context, right? So we'll focus on this question, that loss of TREM2 in mice AD model, it restricts the ability of microglia to surround the plaques, the amyloid beta plaques. In this particular vignette, which is taken from this data, we will use the data that they have deposited publicly from the two mouse, wild type and 5x FAD. We will focus only on the 5x FAD, which is the Alzheimer's version, right? So this mouse already is known to have Alzheimer's, and then we knock out TREM2, and then we notice, or we try to figure out what happens if we knock out TREM2 to begin with. Okay, is that part clear? So there are two versions of the same mouse model. This is 5x FAD because it's an Alzheimer's disease. One of them will show Alzheimer's pathology. The other one is a deficit of one gene, which is called TREM2. We want to see what happens, right? And we're going to start with a cheating operation, which usually I don't recommend. But just because we have limited time, the cheating operation that we're doing here is I'm skipping over a lot of steps, which is actually not a good advice, right? Like ideally I would not do this. The step which is most crucial for any single cell RNA sequencing analysis is QC steps. How do you do quality checks if the data is given to you? The reason I'm skipping over is because, again, I'll have to ask you to download a bunch of files. These are also larger files. It will take time and it's hard to coordinate for one person. So that's the reason I'm going to cheat, but I'm going to be very upfront that I'm cheating in this case. So what I have done is every operation that you would have otherwise have to do to create these objects, I've already done it for you. And I have made this object available on this Google Drive here. So if you click on this link, you will be able to download a RDS object. Now, while I have cheated, I've given you the steps which are actually required to cheat, right? Like which are actually required to create this. And you'll see why I have done this, because otherwise I would have asked you to again download all this data from Geo and then create this object from scratch. But you can use this code in principle to create or to go over and see how this object was created. This is of course conditional on the fact that these files have to exist on your laptop, and that's when you'll be able to create the entire object. So the issue is as follows, that you have different files deposited on Geo. So you can figure out from the file names what they are. This is wild type, this is wild type 2, this is wild type 3. So these are 3 different replicates or 3 different samples taken from the wild type. And then there is TREM2 knockout 1, 2, and 3. And then there is wild type 5x FAD and similarly knockout in all of them. And this is for different stages. We are creating all of them at once. So we will get all the data set itself was done for 2 mouse in 2 different stages. One is at stage 7 and one is at stage 15. This is developmental stage. We will only analyze the stage 15 data because most of their figures focus on stage 15 to begin with. So here is the code that will allow you to create this object. I will not run this code because I don't have all the files right now, and neither will you. But I will walk you over what is the steps required for doing analysis, right? So we are using Seurat. Seurat is a tool that's developed in Rahul Satija's lab at New York Genome Center. And it's a versatile tool to allow you to do a lot of analysis. This is in R. There are equivalent versions or different versions available in Python. It's called ScanPy or ScanP. We will be using R because in this case it works, and it's usually my to-go language right now. So otherwise, if you, again, you're welcome to use ScanP if you want to. The first step that I'm doing is I'm reading this counts matrix. So there's a function. This is from Seurat. We'll see what this function does in just a bit. It reads these 3 different files, right? So the first file that it is reading, for each sample, I'm iterating over different sample names, and I'm reading 3 files. One is the MTX file, one is a cells file, and one is the features file. The MTX file is a matrix format file which stores the counts. The genes files stores the genes, and the cells file stores the barcodes, right? So I will just do this for the sake of showing you what it looks like very quickly. So again, if I blindly copy, I will run into issues. I should be able to read errors, right? So again, it's going to give me errors because a quote is missing. So these kind of errors you'll have to sort of figure out on your own now. Now you're matured, haven't taken this course. You'll be able to figure out what is, so, right? So read what is the error, right? Like, so it told me what is the error. So I could just figure out. This will take a bunch of time, and I'll come back to this in once it is done. But to go back, I read those files. This function will create the counts matrix for you. And the second step that I do is I create a new feature, which is called percent mitochondria. And unfortunately, I did not have time to talk about the biological details about this, which I'm going to do now. Is whenever you are processing any single cell, what happens is there is a dissociation step, if you recall. That dissociation step requires either reagents or you physically dicing the cell so that the tissue dissociates. The tissue has to dissociate before the cells can come out. And in that process, what you're doing essentially is you're stressing the cells, right? You're actually saying the cell, this is stress time because I'm going to remove you from your environment. They will be extremely stressed. Now, when they're stressed, their mitochondrial percentage, which is, you have 2 RNA, you have your usual mRNA, and then what are the other RNAs in the cell? Huh? Louder. MicroRNA. Okay. Fair enough. What other RNAs? Yeah, right. So tRNA, good. tRNAs, rRNAs, and so on. So there is also this mitochondrial RNA, which are not very different. These are except that these are coming from the same. These are coming from the mitochondrial genes. Usually, if the cell is under stress, the RNA will start to decay, and things will start to rupture, and so on. And the cell will start to have more number of mitochondrial RNA reads. These are still poly A tagged, so you can capture them from 10x, and that's what, or any droplet-based sequencing technology. When you do that, if the cell is extremely stressed or it is about to die, right, because you are what you're doing is you're literally hammering the cell in some sense. You cannot draw a hammer, but it looks like maybe like this, right? So if you're hammering the cell is about to die because it's under extreme stress. So all of this will start to decay. There is mechanism in the cell which will cause it to start to decay already. But this is harder to decay in general. The mitochondrial stuff will stick around. So we use mitochondrial percentage, or the amount of the percentage of RNA which is coming from mitochondria, as a QC metric. So if the mitochondrial percentage is very high, this is MT%. If it's very high, which is usually greater than 20%, then it usually will mean that the cell is about to die, right? This is an extremely stressed cell. It cannot handle everything that you have done to it. So we want to exclude these cells which express RNA content coming from mitochondria to be higher than 20%. 20% is an arbitrary number, but it's a number which people use. You can go up to 15%, 5%, and so on, but you need a cutoff there, and that's what we are doing here to calculate for each cell how many amount of what percentage of molecules are coming from the mitochondria. And this is calculated in this column called percentage mitochondria. I will show you once I load up the object. The second analysis, the second sort of step, what I'm doing is, you have gotten all the cells, right? You have, let's say, 29,000 cells. Not all of these will be good quality. They will have different quality metrics. They will have different amount of coverage. They will have different amount of genes expressed and so on. There are two metrics that are often calculated, which is in N count RNA, which is the number of molecules in each cell, and then N feature RNA. None of this is in the slides, so if you have to take notes, please do. Okay, so there are two important metrics for each cell: the number of molecules that it has and the number of features it has. This feature is nothing but the number of genes. So for each cell, I'm basically calculating, if this is your gene, if I calculate the total sum here, let me call it sum C1, this is that number, and N feature, which is the number of features that are expressed, is number of genes with greater than equal to 1 molecule. So even if you cannot see the text, I will just explain what it is, right? So if this is 0, this is 1, and this is 1, then the number of features that are expressed in this cell is 2, because this one is 0, right? So the step that is done is, often you will plot this value across all the cells, and you will get these violin plots. And there are better methods to do it, but usually what you would do is you will try to sort of eyeball what are the outliers, right? So are there cells which have extreme coverage or extreme number of molecules? Let's say this is N count, and now is your exam question. So this is N feature. Okay. Now this is just, there's no math. This is just common. This is just pure logic. If this is the number of molecules that your cell is expressing, and this is the distribution of the number of features that it is expressing, and you plot a violin plot and you see a lot of values here, for example, you will remove this because these are outliers, right? That's hopefully easy to see. In any data analysis pipeline, you'll want to remove the outliers. My question to you, however, is, what would explain this cell? Why does it have so many molecules? Well, the other ones don't. The This cell has high molecules. It also has a lot of genes which are expressed. What does it mean in this case for a gene to be expressed? If a gene does not have, if a gene has more than or equal to one molecule, that is considered to be expressed. When will you see a scenario where a cell, all these dots are at the cell level, everything here that you're plotting is at the cell level. When will it have a lot of molecules? Who said? But yeah, doublets, okay. Huh? It's stressed more, so if it is stressed more, ideally it's possible that you get a lot of debris, but it usually will go the other way around, which is you will have lesser RNA content. So one is technical reason, which is it is a doublet, right? Which is what should have existed in one droplet should have been one cell. It's two cells, and that's why the total number of molecules is larger. Any other reason? That's technical reason. Biological reason. We have seen this. That's possible, yes, right? So some biological change that's happening, it's overexpressing, the entire machinery is just haywire and it's producing more number of things. Larger cells usually will also have larger number of molecules, but in these cases, these are outliers, clearly outliers, because if there are just a few of them, it's very likely that either those are doublets or these are very crazy cells, which at this point we will not want to retain, right? So the numbers that you're seeing here, N count RNA 300 and N count N feature RNA, what I'm filtering is I'm filtering cells which have at least 300 molecules and at max 9K molecules. And this is something that you will figure out by looking at the data. Like these numbers are not panacea. Like these are not which will universally work everywhere. You will have to figure out how these were filtered. The reason I think I chose this is probably because they were filtered on this criteria in the paper. So read the paper, what criteria that they use to filter these cells, and then use that to figure out what kind of filtering you should do. This is something which is not an exact science. There are, again, with this caveat that there are methods to do this exactly. I usually don't use those yet, but I just usually eyeball my data and look at it. I'm not saying that this is the best way to do it, but it's okay to do it, right? It's with this caveat that you understand what you're doing, right? You will throw out a lot of information. If you wanted to be a bit more statistically justified, which I used to do at some point, is you can take 1% quantiles towards each end and just throw them off, right? So you take the 99.5% quantile and 0.5% quantile from the lower ends, throw it out, essentially, right? Because those are your extreme points in the distribution. Okay, and finally, that's what I do. I do it for all the samples that belong to a particular stage. And finally, I can merge them. So at each step, I was reading the counts for different samples, and finally, I'm merging them. I will show you what this object looks like and what these columns do in just a bit. But an object in Seurat is like a data frame. So just like you can assign a column to your data frame, I'm assigning a column of the genotype. What genotype did this mouse come from? So I can figure it out from the sample name, because sample name had either WT, which is wild type, or 5X FAD, which is the other strain of the mouse. So that's what I'm trying to figure out here and putting it in as a separate column. I am also doing the same thing for mouse. Sorry, the genotype here is 2. That will be the genotype here. There is a, I should call it something better. It's the genotype column here captures whether it's a wild type or a TREM2 knockout. Okay, it's not exactly genotype, but just stay with me for that reason. There's a separate column that we add, which is called the mouse type, which is actually technically also a genotype. The mouse type here stores whether it's a 5X FAD, which is the Alzheimer's version, or is it the wild type, right? We will focus only on one of them for now. And again, all of this will become clear once I start it. Any questions about any of this yet? Very quickly, you found out the data from Geo, you downloaded it. you got a lot of files, and with stuff happening in the US, this is always finicky. So it will download at some point. Okay, so I'll come back and show you what the data looks like. But here, the next step that I do is once I get the data, once I get the count files, I prepare an object. What is that object? That object is nothing but you're storing the counts. 29K, gene 1. All you're doing is creating this, which is your counts matrix, and a metadata matrix, right? Now this metadata matrix stores metadata for each cell. So its row names are your cells, and a metadata could be here it's wild type, here it's 5x FAD, and this is your normal wild type mouse, and this is your knockout mouse, and so on. Okay. So there are two objects there are two of these matrices essentially that you're creating in that steps, which we skipped. This step is just involving installing packages that we will be requiring towards the end or during the process. I'm going to exit it because I have them, so I don't need it. But you should install all of these packages. Surat is the main package. Tidyverse, I'm assuming you'll already have. ggRepel is a package for showing name of genes in a plot without overcrowding the plot. glm-gampo is a way to run single cell normalization. I'll talk about it in a bit. Reshape2 is to reshape the files. The other two packages that you're seeing, those are annotations for each gene. We will perform gene ontology enrichment, and it will become clear again why those are required. And the final package is required for doing gene ontology enrichment. I will talk about it in just a bit. Okay, so the first step that I'm doing is I'm reading these objects. What does that object look like? So if you just type object, it will tell you that it's an object of class Surat. It has an assay called RNA, which has 17,953 genes, 27,999 cells, and it has a layer present of counts. Now, at this step, it's very confusing because this is the first time you're touching anything, and it will become extremely dense to some extent. What you can always do is, in R, you always have access to what is the stuff that you're actually storing, right? So you can see here, it's storing this counts as a layer. I had talked about this last time. It's storing something in the cells format. This has something called features where the data is stored and so on, right? So you can actually see at each step what is being stored in your object. So object is called object because it's an R object. And inside that R object, you have these different fields. So I talked about metadata. Those are two different things that you have to store, right? So your counts are stored in this object, right? So it's your entire object mainly consists of these things, right? So this is what your counts matrix looks like. It's a sparse matrix. Row names are genes, column names are cells. You can do DIMZ. This is 17,000 genes, 29,000 columns. You also have meta, which is the metadata, and you can, again, you might not know what it is doing, but you can always, again, use @operator to figure out what is stored in each of these objects. So it stores two things: assays, which I showed you just in the previous step, and the next thing that it stores is this metadata. So I'm just doing metadata now, and I can do this, which I told you there. The row names are now cells, and the metadata stores what all things are there. It stores this N-count RNA, which is the total number of molecules in each cell. This is the cell. It stores the total number of features that are present. It stores the percentage mitochondrial content. And it has other columns that I had added: sample name 2, and genotype, and mouse model, and so on. Okay. So if you go through the tutorial for Surat, which I'm going to provide you a link to, so that if you are curious, you should be able to run it on your data set as well. You can always plot these features. I mentioned that in an ideal world, you will plot them first to see what is the distribution of these two quality metrics in your data. So the way you do this is there's a function called VLN plot, which does a violin plot. It takes its first argument as object, and the second argument is a list of features that I want to plot, right? So that's what the plot would look like. Now, each of this row, the x-axis that you see is the different sample that I had processed this with. This is the total number of molecules. You will see that the distribution is somewhat slightly higher on this side, but all of this is capped here into at 300 because I had subsetted this object already, right? You can see that this distribution is somewhat chaotic, but overall it doesn't have extreme outliers. Maybe you can argue that I should have removed that particular point, but again, some of this is a bit subjective. You can always come back, and whatever analysis you do, you can always come back and say that whatever am I observing, is that something that I could explain based on this technical factor? So at this point, there is not necessity of you really going, sort of berserk on, no, I want to get this right. You should try at some point, but it's very subjective. So at some point, you just have to accept what you have. At N feature RNA, also you will see, right? We have how many genes in the matrix? So 17,000-ish. What is the average number of genes that are being detected? Exam question. On an average, how many genes are you detecting? Huh? 1,000-ish, right? 1,000-ish genes, right? 17,000 genes, but on an average, in each cell, you're only detecting 1,000 genes, right? That's extremely sparse. This is where the sparsity comes from. Even if you look at the total number of molecules that you have, the total number of molecules are what? Usually, what will be the average here? Yeah, bit less than 3,000. It will be like 2,500. Again, I don't know the answer. So if you don't know the answer, you use computer. Yeah, I just ballparked. I just looked at the plot. Guesstimation. 1,000. So you have like a lot of distribution here, right? So 1,000 is the upper cap of the total number of molecules you'll see. The average will be somewhere in between, maybe 700 or something like that. Okay. This is something which you would do. For example, the median coverage for N-count RNA is actually very small. It's just 1,000, right? This is the total number of molecules. It's just 1,200 molecules per each cell. And the number of genes detected is 600. That's it. So 600 genes per cell overall, right? Very, very small number. But you can still do biology with all of them, right? Like you can still, even if you're measuring 600 genes on an average in each cell, you can do very interesting biology overall, and that's what we're going to do now, right? I just want you to be able to understand and appreciate the scale or the downscaling of the data that has happened. From 17,000 genes, on an average, we are able to only see 600 genes per cell, right? This number is 600. And still, we will be able to draw a lot of insights from this data. Okay? Okay, we'll proceed. The first thing that we want to do is, I ask you to explore the objects and so on, which I'm going to do with you. I had started this last time. I'm going to just do it here again. So the first thing is this counts matrix. You can load up your counts matrix, which looks like this. One second, So there's some subtlety which you don't have to worry about, because there are better ways to get this data, and this at this point it gets slightly technical in which how the data is stored, right? So there are two ways to access this right now. Well, this is not the right way to access the data that is stored in Surat. but I will skip that discussion because we don't have time. But usually what you can do is, right, so if you don't know what is going on, right, so Sirat, get counts, right, so somebody would have already searched it, and there's a list of commands which are already given here that you will be able to analyze, how to access features, how many number of cells are there, how many number of features are there, and so on. So it's a very, very well documented package. Overall, it has a lot of what's called vignettes. This is the tutorial that I would strongly recommend that you do, and is usually recommended to anyone who's entering the field to get an idea of what the analysis looks like, right? The tutorial that we have is slightly advanced than that particular tutorial because we're going to do some more downstream things, but it's designed on a similar lines, right? So what I was doing was I took a tangent to explain the differences between these two things, right? So in this case, I am directly accessing the counts as a attribute, and you will notice the difference, and you will tell me what is the difference between this and this. What is the difference, if at all? I'm accessing the counts in two different ways. Difference? Okay. Yeah, that's right. So you don't know actually anything at this point. It doesn't have the features. It doesn't have the name of the features, and it doesn't have the name of the barcodes. The first one does, right? The dimension of both of them is the same. Okay. So we should have worked with this data. I should have updated the version to, in fact, reflect this, but I'm going to do it right now. You can read about how the data is stored and what are the differences between those two slightly later. I'll post links to read about how to actually access data. Okay, but this is what my counts matrix looks like overall. Now, once you have access to this count matrix, we can know the first question that I would like to know, for example, is how many number of zero entries that we have, right? So last time I talked about that. Let me show you something, which is, this is the amount of bytes that this object takes when it is stored in memory. Okay, this is around whatever is the number. Let me divide it by 189. This is 0. This says 0.2 bytes, but it is actually, let me do it this way. Yeah, this is like 226 whatever KB or MB actually, right? No, KB. So if this is 226 KB, this is stored in I will not do this mistake. So one second, let me do counts sample. So what I want you to show, what I want to show you is that the format that it is stored in is very, very important, right? So we'll just take, it's already having trouble, 1000 genes and 1000 first cells. Okay, so I am already running into issues. Shouldn't happen. Okay. Okay, I'm going to just restart my session.